I'm Grace.
And I'm Adam. We're biology educators here at SchoolOnX, and we are so excited you're joining us. Today we're going to go on a field trip to probably the smallest factory you could ever possibly imagine.
That's so true, Adam. We're not just going to talk about biotechnology. We are literally going to build something amazing with it, piece by piece. Okay, so to get us started, I want you to picture this. Your entire genetic code, all 3 billion letters of it, is one gigantic library. And inside that library are thousands upon thousands of books. And these are the recipes for every single protein that makes you, well, you. So here is the challenge. You need to find one recipe. Not a chapter, not even a whole book, just one single recipe for, let's say, insulin. How in the world do you find that one page in a library that big, copy it without a single mistake, and then make millions of perfect copies? I mean, honestly, it sounds like something straight out of science fiction. You're right, it absolutely sounds like science fiction. But Adam, that seemingly impossible task is exactly what biotechnology allows us to do. And the key to really getting it is to stop thinking like librarians searching through a giant library and start thinking like engineers.
Oh, I love that. So we're not just looking for a recipe. We're actually going to design and build a microscopic factory completely from the ground up. A factory that can produce any protein we want on a massive industrial scale.
Right, so before we put on our engineering hats, let's just make sure we're all on the same page with what biotechnology even is. Now, there's a formal definition from the European Federation of Biotechnology, something about integrating natural science with organisms and cells, but what does that actually mean in simple terms?
It means we're basically hackers. But instead of hacking computers, we're hacking life itself. We're taking living things, everything from tiny little yeast to complex cells, and using them as tools to make stuff that we need. And, humans have been doing this for thousands of years with things like bread and yogurt. The only difference is, today, we're doing it with unbelievable precision to create things like life-saving medicines and vaccines.
Okay, Grace, so every great factory needs a great blueprint. For our microfactory, that blueprint is the specific gene we want to use, our gene of interest. So I guess our very first job as engineers is to get that blueprint out of that massive DNA library. Exactly. But this is incredibly delicate work. You can't just go in and rip the page out. We have to find the exact gene and cut it out with surgical precision without damaging a single letter of the code. And to do that, we need some very, very special tools. Okay, so our first tool is, and I'm not exaggerating, one of the coolest things in all of biology, a pair of molecular scissors. These are enzymes, and they're called restriction endonucleases, and they are just mind-bogglingly precise. They don't just snip DNA randomly. Each one is programmed to search for and cut only at its own unique specific sequence of DNA letters. The very first one discovered, HIND-2, completely changed the game, and today we have this incredible toolkit of over 900 of them, each one a specialist for a different job. So how do these tiny scissors know exactly where to make the cut? Well, they're looking for a kind of secret code word. It's called a palindromic sequence. Now, you know what a palindrome is in language, right? It's a word that reads the same forwards and backwards, like the one you see here, Malayalam. Well, in DNA, it's a sequence that reads the same on both strands when you read them in the same direction, let's say 5' to 3'. A classic example is GAITC. Now, if you look at its partner strand, it reads CTTAG, but if you read that partner strand from 5' to 3', what do you get? GAITC. It's a perfect match. And that is the target. But here's where the real magic happens. Take a look at how this enzyme, ECRI, actually makes its cut. It finds that GAATC sequence, but it doesn't cut straight through the middle like a regular pair of scissors would. No, instead, it makes this staggered cut between the G and the A on each strand. This is so incredibly clever because it leaves these little single-stranded overhangs. We call them sticky ends, and believe me, that name is absolutely perfect. They are the key to our entire factory assembly line, which we're going to see in just a minute. Okay, Grace, that is seriously brilliant. So we've used our molecular scissors, and we've successfully cut out our blueprint, our gene. But a blueprint just sitting on the table is pretty useless, right? It needs to actually get to the factory's assembly line so it can be read and used. Precisely. We need a way to transport it. We need basically a microscopic delivery truck. So in biotechnology, our little delivery truck is called a cloning vector. Your textbook uses this fantastic analogy. Think of how a mosquito can carry the malaria parasite from one person to another. Well, our vector does the same thing. It carries our precious gene of interest into our factory worker, which is the host cell. And the most common vectors we use, discovered by scientists like Stanley Cohen, are called plasmids. These are these amazing little circles of extra DNA that bacteria just have hanging around. The cool thing is they copy themselves totally independently of the main bacterial chromosome, which makes them the absolute perfect vehicle for our blueprint. So what makes for a good delivery truck? Let's take a look at a classic model, the PBR322 plasmid. Every good vector needs a few essential features. First, you see this part here labeled ORI? That's the origin of replication. You can think of it as the ignition key. It's the signal that tells the host cell, hey, you need to make copies of me. Second, it needs something called selectable markers. On this plasmid, we have AMP-R and TET-R. Think of these like special license plates or barcodes. They give the bacterium resistance to certain antibiotics, which, as you'll see, is going to be incredibly useful for helping us find the cells that actually have our plasmid later on. Okay, Grace, I have a question, and I bet our students are thinking the exact same thing. All of this is happening at a microscopic level. How in the world do we know that our gene actually got into the plasmid? I mean, we can't just look through a microscope and see it.
That is the million-dollar question, Adam. And the answer is this incredibly clever trick called insertional inactivation. So here's how it works. Remember that barcode, the TET-R gene that gives resistance to the antibiotic tetracycline? Well, we intentionally use our molecular scissors to cut the plasmid right in the middle of that gene. So what happens when our gene of interest inserts itself? It breaks the barcode. The TET-R gene gets scrambled and it's totally useless now. So our new recombinant plasmid still has ampicillin resistance, but it has lost its tetracycline resistance. So by growing the bacteria on different plates with different antibiotics, we can do a little detective work to find the exact colonies that survived on ampicillin but died on tetracycline. And those are our factories.
Okay, so we've got our gene blueprint with its sticky ends, and we have our opened up plasmid vehicle with its matching sticky ends. And just like two perfectly matched puzzle pieces, they're naturally drawn to each other and pair up. But that connection is only temporary. To make it permanent, to really seal the deal, we bring in our final essential tool, an enzyme called DNA ligase. This is our molecular superglue. It forms a permanent, strong chemical bond, sealing the backbone of the DNA and locking our gene into the vector for good. And just like that, we've actually done it. Let's just take a second to recap what this diagram is showing us. We took a piece of foreign DNA, that's our blueprint. We took a plasmid vector, our delivery truck. We used the exact same molecular scissors on both to create those perfectly matching sticky ends, and then we used our molecular glue to join them together. The result is this beautiful hybrid molecule, a recombinant DNA molecule. You know, this is the groundbreaking work that Herbert Boyer and Stanley Cohen figured out back in 1972, and it basically launched the entire field of modern biotechnology. Our instructions are officially packaged and they are ready for the factory floor.
All right, team, this is the moment. The blueprints are designed, the delivery trucks are loaded up and sealed. It's time for the main event, getting these instructions inside our factory worker, the host cell. And usually that's a simple, reliable bacterium, like good old E. coli.
And this is actually a bigger hurdle than you might think. See, DNA is hydrophilic, which means it loves water. But cell membranes are made of lipids, or fats. And as we all know, water and fat just don't mix. We can't just knock on the bacterium's door and ask it to please let our plasmid in. We have to persuade it. Let's say, forcefully.
So the way we persuade them is by making them competent to take up DNA. Think of it like this. First, we give the bacteria a cold bath in a solution with calcium ions. This chills them out and is thought to poke these temporary, tiny little holes in their cell walls. Then, we mix in our precious recombinant plasmids and keep the whole thing on ice. Now for the most important step, a sudden, really brief heat shock. A quick blast of 42 degrees Celsius heat, and then boom, immediately back into the ice. This crazy change in temperature creates a pressure difference that literally shoves the plasmids through those little pores and into the cells. It's a little bit brutal, I'll admit, but it works really well. Woohoo! The blueprint is in. Our very first microscopic factory is officially built, and it is ready to go. But let's be realistic. One single tiny bacterium isn't going to produce enough insulin to help even one person, let alone millions. The real power, the real magic of biotechnology is all about scale.
That's exactly it. We need to go from one successful factory to a whole army of billions of them, all working around the clock, just churning out our protein. And for that, we have two incredible techniques for mass production.
First up, what if we just need more copies of the blueprint itself? Let's say we started with only a tiny, tiny scrap of our original DNA. We can amplify it, or make more copies, using a revolutionary technique called PCR, which stands for the polymerase chain reaction. PCR is a scientific miracle. It's basically a molecular copy machine that can take a single piece of DNA and in just a couple of hours make a billion copies. That's a 1 with 9 zeros after it. It's just breathtaking.
And it does this through a really simple, repeating cycle of just 3 steps. First is denaturation. You heat the DNA up to about 95 degrees Celsius, and that separates the 2 strands. Second, annealing. You cool it down a bit so these small guide molecules called primers can attach to the strands. And third, extension, a special heat-stable DNA polymerase enzyme, which originally came from a bacterium called Thermus aquaticus that lives in hot springs. It builds a brand new complementary strand. You just repeat this cycle 30 or 40 times, and just like that, billions of copies.
Okay, so just so we're all clear, PCR is for making more copies of the DNA blueprint itself. But now, we need to make more copies of the factories, the actual bacteria that are holding our blueprint. And for that job, a little petri dish in the lab just isn't going to cut it. We need to go industrial.
We do. And for that, welcome to the world of bioreactors. These are basically giant, sterile, high-tech industrial vats. Some of them can hold up to 1,000 liters of culture medium. They are custom built to be the perfect 5-star hotel for our bacterial factories to grow and multiply in.
And as you can see from this diagram, it is so much more than just a big tank. A bioreactor is a perfectly controlled microenvironment. It has a powerful agitation system, like a blender, constantly stirring the mix. It has an oxygen system, bubbling in fresh air. It has sensors and controls for the perfect temperature and pH level. It has sterile ports to add fresh nutrients or take out samples. Its entire purpose is to keep the cells in their happiest, most productive growth phase so that we can maximize the amount of our protein that gets made. It is the ultimate factory floor. This is just incredible, Grace. So inside that 1,000-liter tank, we have trillions of bacteria, and they are all producing our protein. We did it! The factory worked! But wait a minute. We can't just bottle this bacterial soup and give it to a patient, can we?
Absolutely not. Adam, you just hit on one of the most critical, most complex, and honestly most expensive parts of the entire process. The manufacturing might be done, but now the really hard purification work begins.
This stage is called downstream processing, and it is a massive undertaking. First, you have separation and purification. We have to crack open all those bacteria and somehow fish out our single target protein from this complex soup of cellular junk and waste products. Then we have to purify it to an unbelievable standard of purity. After that, it gets formulated with preservatives to make sure it stays stable and effective. And of course, for any new drug, it has to pass through years of rigorous clinical trials to prove its safety. And finally, every single batch goes through strict quality control checks. The big takeaway here is this: making the protein is only half the battle. Purifying it safely is the other, equally important half. And that really just perfectly summarizes the entire incredible journey. From that first clever little snip of a molecular scissor, all the way to the final quality control check on a perfectly purified vial of medicine. This whole factory process is what makes biotechnology so revolutionary. And as your textbook puts it so well, it has brought about a true qualitative improvement in health and food production. This isn't just a theory in a lab. It's a powerful engine for creating real-world, life-saving products that help people every single day. So today we walked you through the entire blueprint for building one microscopic factory. But the amazing thing is, the fundamental principles are exactly the same whether you're making insulin, a new vaccine, or maybe even an enzyme that can break down plastic waste.
The possibilities really are limited only by our imagination. So we want to leave you with this challenge. Now that you know how the tools work, if you could build your own microfactory, what world-changing product would you design? Really think about it. From both of us here at SchoolOnX, thank you so much for building with us today. We are so proud of what you're learning. Keep exploring.